Hello everyone, good morning and welcome to Grade 7 Science. Let's put our knowledge on the scientific method to a realistic example that includes some of the terms you'll be needing to use and understand. John watches his grandmother baking a bread. He asks his grandmother what makes the bread rise. Her mother explains that yeast releases a gas as it feeds on sugar. John wonders if the amount of sugar used in the recipe will affect the size of the bread loaf. John researches the areas of baking and fermentation and tries to come up with a way to test his hypothesis or question. He keeps all of his information on this topic in a journal. John talks with his teacher and she gives him an experimental design diagram to help him set up his investigation. After talking with his teacher and conducting further research, he comes up with a hypothesis. If more sugar is added, then the bread will rise higher. Hypothesis is an educated guess about the relationship between the independent and dependent variables. Did you know the difference between the independent and dependent variables? The independent or manipulated variable is a factor that's intentionally varied by the experimenter. For this experiment, he's going to use 25 grams, 50 grams, 100 grams, 250 grams, 500 grams of sugar in his experiment. A dependent variable or responding variable is a factor that may change as a result of changes made in the independent variable. In this case, it would be the size of the loaf of the bread. His teacher helps him come up with a procedure and list of the needed materials. She discusses with John how to determine the control group. In scientific experiment, the control is a group that serves as a standard of comparison. The control group may be a no treatment or an experimenter selected group. A control group is exposed to the same conditions as experimental group, except for the variable being tested. Remember that all experiments should have a control group. Because his grandmother always used 50 grams of sugar in her recipe, John is going to use the amount in his control group. John's teacher reminds him to keep all other factors the same as any observed changes in the bread. It can be attributed to the variations in the amount of sugar. The constants in an experiment are old factors that experimenter attempts to keep the same. Can you think of some constant for this experiment? That might include other ingredients of the bread recipe, oven used, rise time, brand of ingredients, cooking time, type of pan used, air temperature and humidity, where the bread was rising, oven temperature, age of the yeast. John writes out his procedure for his experiment along with a material list in his journal. He has both of these checked by his teacher, where she checks for any safety concerns. Trials refer to the replicate group that are exposed to the same conditions in an experiment. John is going to test a sugar variable 3 times. John comes up with a table that he can use to record his data. John gets all his materials together and carries out his experiment. John examines his data and notices that his control worked the best in this experiment, but not significantly better than 100 grams of sugar. John rejects his hypothesis, but decides to retest using sugar amount between 50 grams and 100 grams. Once again, John gathers his materials and carries out his experiment. Here are the results. Can you tell which group did the best? John finds that 70 grams of sugar produces the largest loaf. His hypothesis is accepted. John told his grandmother about his findings and prepared to present his project in science class. Okay, we are here to discuss about a scientific inquiry and scientific method. A scientific inquiry is a process used by the scientists to research a question, while a scientific method is a step-by-step process of solving or answering the question. There are 6 major steps in scientific method. Begin with observing, to be followed by identifying a problem, formulating a hypothesis, conducting an experiment, making a conclusion, and establishing the results. Observation or observing is any information you obtain with your senses, such as what do you see, what do you hear, what do you smell, what do you feel, and what do you taste. There are 2 types of observation. First is the qualitative observation. These are the observed characteristics such as the color or smell, such that if you see an gumamela, it's color red. Or if you smell something bad, that is a qualitative observation. While a quantitative observation is any observed characteristics in measurement, such as how many or how much, like how many train go in a particular street, what time. It can be a quantitative observation. After knowing or observing, we need to identify a problem. Identifying a problem, we need to state the problem as a question. We make it as specific as possible. Make sure that your problem is testable. It can be done by a simple or complex scientific method or an experiment in order to prove your hypothesis. Next part is formulating a hypothesis. A hypothesis is a proposed answer to a question. It is sometimes called an educated guess. It is a temporary answer to a question, a guess to a problem you pose during the observation. There are 3 ways in stating the hypothesis. The first one is a null form. A null form states that there is no significant difference between the variables tested, or some sense between the observable traits or characteristics you've mentioned. For example, there is a varying salt concentrations will not affect rice seeds germination. We always state the null form of hypothesis into a no significant or negative statement. The second one is the alternate form. It states that there is a significant difference between the results of variables being tested. Take for an example. Varying salt concentrations will affect rice seeds germination. It's a positive statement. Next, the last one is the if and then statement. It states that if a certain conditions cause is applied, then a corresponding observation effect already occurs. For example, if we try to add the varying salt concentrations, then there's an effect in rice seeds germination. So that's how we state a hypothesis in experimental setup, or what we call scientific method. After we formulate our hypothesis, we need of course to test our hypothesis using a very well designed experiment. We need to have a method in order to prove the hypothesis whether it's correct or incorrect. Whether the hypothesis we proved during the experiment is correct or incorrect, we need to establish the results. It is still a part of scientific community, whatever or regardless happens during the experiment. There are components. First one is what we call variable. It is a factors that can be changed in an experiment to see the effects. We have independent or the manipulated variables. It's a factor that cause changes. A dependent or responding variables. A factor that changes in response to the manipulated or independent variables. A control group. A standard by which any change can be measured. An experimental group or a group or population that tests to see the changes. And a constant are the factors that are kept the same all throughout the experiment to see any changes in our setup. Making a conclusion. We need to state our conclusion based on our data by displaying our data through graphs, line graph, bar graph, or any type of graph and interpret it. In making a conclusion, always make sure that the conclusion are based on the data obtained all throughout the experiment process. And it can prove your hypothesis or lead to another, which is a scientific idea. And lastly is we need to publish our results. Publishing our results includes presenting our findings in a scientific community, publishing it in the books, journals. You also need to have a scientific fair or science fair on the academic year, which can be expressed all throughout your presentations, the experiments you've done all throughout using a scientific method.